The trial comparing lenvatinib to sorafenib was meant to first demonstrate non-inferiority, so meaning that to demonstrate that the drug effects were similar to those achieved by sorafenib. And in a second watch, they also tried to demonstrate that the drug was superior to sorafenib. So the first analysis was positive. Therefore, they demonstrate that the patients treated with lenvatinib has similar survival to sorafenib. I think in the trial, the results was around 13.5 months for lenvatinib and 12.5 months for sorafenib. Lenvatinib in this study show that the toxicity is relevant but manageable. So there were not remarkable treatment-related deaths reported in the study. And the important additional information I would say for this trial is that the treatment achieved objective response in 25% of the patients, which is quite remarkable in Frontline, whereas for sorafenib is less than 10%. I think that this difference is appealing, particularly because when you are treating a patient, it's always good to show objective responses. This means that the tumor is shrinking.
Here at ASCO GI, we presented the results of the central independent blinded read of the all imaging data collected for the REFLECT study. These included an independent blind assessment performed according to standard RECIST 1.1 and according to mRECIST. The bottom line is that PFS and TTP are almost the same between standard and modified RECIST, and almost the same as the one reported for the local assessments done at the sites. This is not surprising because the main difference between standard RECIST and modified RECIST is in the measurement of the target lesions, and therefore this will mostly impact objective response rather than TTP or PFS. Now, for objective response, the central blinded read had confirmed that with the use of mRECIST, you can capture 2 times more responders in either treatment arm, so both in sorafenib and lenvatinib, and also confirmed that lenvatinib had significantly higher response rate than sorafenib. So now a key question is whether response by mRECIST is a true, reliable surrogate for overall survival. So in other words, whether responders truly have improved survival and clinical outcomes than non-responders, as suggested by prior investigations. This is very important information that hopefully will be available in the near future, because this new information could potentially be very helpful in the clinical decision-making process for clinical practice.
The recent report in this ASCO GI meeting, what is addressing is that in the general trial, they were reporting the response rate based on investigator, and in this study they are reporting the objective response assessed by central radiological review that generally is more precise. And the data is quite shocking in a sense that the objective response by modified RECIST reaches 40%. This is unheard of for a TKI in HCC and is giving you an idea of how potent is this drug in terms of achieving anti-tumoral effect. And this has also been reported in other tumors with the same drug, lenvatinib, in renal or thyroid cancer.